So what I'm going to be talking about is a particular project. I mean, at Zifo, we've worked with both large and small companies on digital transformation. As a consulting organization, we don't name our clients, so I'm not going to say who this is with. You might be able to have good guesses. But it is a global top 10 pharma company. So the problem statement really is that the development sciences organization is very siloed. In process chemistry, in process engineering, they've been using the Cambridge Soft ELN to capture data. They're actually in the process of moving to Dotmatics for that. And then within the analytical R&D, there are actually two IDBS ELNs, one validated, one non-validated. There's also a separate drug product development IDBS instance, which is pretty much used as paper on glass historically. And then the DMPK bioanalysis group have a fourth IDBS instance. And the teams can't see each other's ELNs. So the data very much lies within that organization. LIMS, similar structure, the ARD, analytical R&D, have a sample manager instance, and Sapio exemplar is used in DMPK. So there's a really strong business case for making the data fair, which at the moment or historically it really hasn't been. So this project is kind of complex. There's obviously the sponsor company who provide overall leadership. They provide the SMEs, the use case leads, etc. There's then another third-party consultancy who have sort of got the project lead for this, and they've also done some of the sample manager analysis. Zifo has provided a lot of services, consulting, business systems analysis, IDBS template development, Sapio workflow development, etc. There's then another consultancy company who have done Pega development, which is used as a request system. It's a low-code BPMN development system. And then yet another company who provided an overall project manager. So overall, to date, there's over 100 individuals who have been involved in the project, so it's clearly pretty large. So the dev science space is kind of complex. There's lots of dimensions. So we've got small molecule and biologics to be taken into account. We've got drug substance and drug product. We've got early and late stage development. We've got both CMC and DMPK, although I'll be concentrating on the CMC. And then there's GMP and non-GMP. So the way that we have approached this is a use case-based approach. And the idea is that you can't solve everything at once. Digitization has been said many times is a journey, and you need to start somewhere. So the idea is the use cases are essentially stepping stones, and then over time they can be grown upon, and you end up with a bridge covering the whole area. So in terms of the overall digital journey, and this is probably there's been various versions of this slide at various meetings, but this is kind of our version. So the current state is that some of the data is sort of fair. There's some automation, but it's really an electronic organization. The data is being captured, but it isn't really digital. Where we're trying to get to is the situation where virtually all the data is fair. We're ready for the AI/ML approaches, but not necessarily doing them yet. And that's essentially where we're trying to get. So, and the reason we're doing that is you can't really start doing the artificial intelligence until you've got fair data. So the first step has to be getting to there. And currently, like I said, we're not really meeting the FAIR criteria. So the scope is to get there, and then that will set the stage to start building the knowledge graphs and doing proper AI approaches. So where do you start? How do you build a use case? So obviously you want to go for the use cases with the highest business impact. So there are a number of ways of assessing what is high business impact. You obviously want to look at workflows that are run a lot. It's pointless starting with a use case that you might run once every couple of years. You want things that are run pretty continually. You also really want to look at the use cases where you have a real need to visualize the data or to look at the data, analyze the data. So that's one of the other things. You also want to look at use cases where there are pain points, and the pain points particularly around data access, data accessibility. And we've found that a lot of those pain points are where different swim lanes are interacting. It's the fact that you've got siloed data and you're trying to get data from one swim lane into another swim lane. So you really want use cases that span multiple groups. And then there's also corporate politics, and that this company have got development sites in the US and in Europe, and you don't want to just do one. You need to be inclusive. So we've made certain that use cases cover the various sites. So the next thing is about data digitization. It's not about process optimization. We work on the assumption that the scientists know what they're doing. They've got pretty well optimized procedures, and we're not going in looking at changing the way they do the science. What we're doing is making certain that we're capturing the data in a way that is fully digital. So the process changes will be minimal, but what they do is try and eliminate or at least reduce manual data entry, and particularly repetitive data entry. I mean, in terms of data integrity, you've got a real problem every time you've got manual data entry because transposition errors are notoriously high, particularly with numerical data. The other thing you need to do is know when you've done. There's always the chance of scope creep, and the business always want more and more features. We need to make certain that we define clearly what the scope of the use case is and deliver the key functionality rather than all bells and whistles. You also need to bear in mind that this is going to be an ongoing process. There's always going to be new technologies, new equipment, etc. And you need to have a framework that allows you to onboard new technologies as they become available. You don't want to be stuck with 2022 technology when it's 2027. So for that, you need a good data governance team that will ensure that there's continued development. It's not just a journey for the duration of this project. You need a long-term commitment to digitization. So historically, IT projects have always been very much driven by systems. This project really turns it on its head. So the main driver is the science. We're looking at what is the science that's being done, what's being captured, who needs to use the data. And from that, you can then get a data perspective. And only once you've got the data perspective can you actually start looking at the systems that are involved. So for the benefit of time, I'll not go through the bullets on this. But what we've put in place is a playbook for how to do digitization. So there are a number of steps. The first is you need to validate the use case. You need to look at through two lenses. Historically, we've always looked at the people generating the data, but we also need to look at the perspective of the people who are doing the filings, who are going to use the data. And they've largely been neglected. I don't know how, I can go backwards. I think I did a double. Okay, so I will go through yeah. So the first thing you do is evaluate the use case. So you're looking at improving data quality, increasing efficiency, and in development, the overall timeline is pretty much determined by the clinical cadence. Clinical trials determine how quickly you get a product to market. So we're probably not going to be able, with a CMC approach, to reduce the overall time to getting a drug approved. But what we can do is increase the throughput. We can allow, with the same number of resources, to do maybe twice as many projects. And that's what we're trying to solve. Once we've done this, we get review from senior management to check that they're on board with putting the resources up for doing the digitization of that use case. We then map out the current state and map the pain points, and then envisage a future state where we can reduce those pain points. So as I said, we need to then look at the data operations that we're doing at each of those stages in the future state. And we create a wireframe for each of those. It's not a user experience exercise, it's a data exercise. We're checking that we know what data we need to be able to update or receive for each of those stages. And from there, we go on to creating the user stories or requirements and starting the implementation. So one of the things that's important is we went from the perspective that no one really understands the full complexity of the data model within the organization. So we're kind of building the data model as the use cases grow. So we started off with a fairly sort of simple, well, you've got materials that are the concepts and you've got batches that are instances. You've then got samples that are based on the batches, and then you can have analyses done, so you've got tests and results, and green here is what we make, blue is what we do, what we analyze. We brought in a request system, so we can request samples or we can request tests. We brought in processes, so that processes turn one batch into another or one material into another. And then for results, tests, and processes, we have parameterization. So what we've done is got a data model that's very flexible. We have a relatively small number of different parameters, but we can have context attached to those parameters that will determine, if a parameter might be concentration, the concentration can be an IC50 value, it can be a concentration of drug product in a vial or whatever. So the context allows us to work with a manageable number of different parameters. So we then look to build a minimal viable product. It's got to be useful to the team, and we try and build that in sort of 6 months kind of time frame. Again, I'll move on. So I just want to focus on one example of a use case. And this is in the late drug substance space. So, and particularly around process control justification. So when you're making a drug, and this is applicable to small molecule drugs, you need to understand the parameter space, things like the temperature, pressure of reaction, stir rates, time and amount of additions. And from that, you can then put your control strategy in place to say, as long as we're within these parameters, we know that our purity profile is going to be consistent. And before digitization, what was done is they'd run a design of experiments to define the parameter space. They'd then perform different reactions for each of those parameter sets and do HPLC to measure the purity. And then they would capture for each reaction step, they'd use Excel to capture the data. And it was each was a very large Excel file, and then trying to collate those through an overall control strategy was virtually impossible. So what we've done since then is try to automate this in some way. And the solution we've put in place is the reactions are done using the Metal Toledo system of Optimax and EasyMax reactors. They generate an XML file with all of the details of temperature, stir rate, etc. We send the samples to HPLC, and then we use Luminata from ACD Labs, which is used for tracking impurities. And they've already built an integration with the iControl XML. We've got an existing integration to bring in power data in. So we can then use Luminata to curate that, and we then push that to a data warehouse and use Spotfire on top of that to visualize. And what we've done in this way is, compared to just using the reactors but using the Excel file, we've taken essentially about an hour off each reaction. And given that there can be hundreds of reactions or hundreds of parameter sets for each reaction step, that's quite a time saving compared to what they were doing before using the EasyMax. It's about 3 hours per reaction. So just to summarize, digitization is very much a journey. You need to get commitment from the team members. You need to identify the quick wins that are going to deliver value to the business. And importantly, given that it's a journey, you need to deliver regularly. To get continued engagement, you have to continue showing value to the business in doing this. There are some advantages of the use case approach. Any one team for a use case is only needed maybe for a year, even though it might be a 5-year overall project. We can then dynamically direct resources to where the need's greatest. And as I said before, we don't need to understand the data model from the start. There are some gotchas there. You need a good data architect behind it. You've got to limit scope creep. You need good, strong use case leadership and consistency in that. And as was said in several talks, you don't want to create mud. You want a data lake that is usable. So you don't try and capture too many details. You really look at the lens of what are the data attributes that you're going to re-look at? What are the things you need to visualize or report? And just capture those rather than capturing everything. And the other thing is communication amongst the teams. It's very easy to under-communicate. So I will leave it with an acknowledgement slide. It's been a large team with a number of people to preserve anonymity. I can't thank people individually other than the Zifo team that have been involved there. So thank you.
The other thing I would say is when you had your slide of where we're all from, you didn't have New Jersey up there. And New Jersey is my home state, but it's also, in its heyday, was considered the medicine cabinet of America. It's where all the big pharmas had manufacturing and R&D. And I was privileged, really, to grow up in New Jersey as a chemist because I learned a lot working in the industry and then joining Waters in 1984. But when I was a chemist, and I'm humbled actually to be talking about digital transformation because as a chemist at Ingridion back in those days, how many of you know what a planimeter is? Does anybody know what a planimeter is? It's a device that you would roll over a chromatogram from a strip chart recording to measure the area under the peak. And then you would literally cut the chromatogram out and weigh it. And that's the technique that I use to characterize polymers at Ingridion. So to be standing here talking about digital transformation and data mobility coming from a humble beginning of literally weighing printouts in order to get quantitative data it's a testament to how technology has evolved for sure. So achieving data mobility using a scientific integration platform, which is really an integration platform as a service. And we're a relatively new company. September 3rd marked our 3rd year. We're getting a lot of traction, and I couldn't explicitly give you a use case. We're working on one which I'll reference at the end of my presentation, but the attributes and the challenges that are faced in the lab when trying to connect the silos that so many of you have talked about and are challenged with integrating is a problem. And you really can't have digital transformation unless you connect those silos. So some of the attributes, well, the need at the high level is connecting the science, the data, the instrument, the devices, connecting really the silos in a manner that's DI compliant. Particularly with the regulated space, everything needs to be compliant with the FAIR principles referenced many times. But the attributes of data mobility and what we call peer-to-peer concepts starts with mobilizing data from all the endpoints. And the endpoints are the silos that you've been referencing, no matter what they are, whether they're complex instruments, devices, or simple devices like titrators, balancers, pH meters, particle counters, or sophisticated systems like MES, LIMS, ELN, data lakes, et cetera. They're all endpoints that ultimately need to be connected. Data aggregation as a principle is not a requirement for data mobility. A lot of people think you need to collect all the data in order to get it moving to where you need it, when you need it, and in the right shape you need it in. That's not a prerequisite. This notion of peer-to-peer connectivity of endpoints is somewhat new, and it's different because the next slide I'll talk about connecting 2 points together is simple. It's been done for probably 20, 25 years. But doing it in a manner that allows configurability and flexibility and dynamically being able to edit the flow of data is somewhat new, and we'll talk about those concepts. What it means in lab context is lab resources can easily exchange data with multiple resources. So you're no longer in a point-to-point scenario, you're in a one-to-many or a many-to-many scenario of moving the data where it needs to be. Data arrives at its target destination at the appropriate time, in the appropriate format, and in context. I've heard that word used several times from the previous speakers. Very important to kind of put structure around the data. And then this is a concept. There's no data consumers or data generators. In previous lives, the slide I'm going to show next was referred to as data generators being lab equipment, data consumers being things like LIMS or ELNs or MESs. The reality is both can consume and generate data, and both need to be connected in a manner that allows that easy exchange of data. So the challenge that the labs face is these are the silos that was referred to earlier. You've got an eclectic mix of diverse instruments, very heterogeneous in their file formats, typically proprietary binary formats. You've got what historically was referred to data generators on the bottom. These might be your standalone devices, as mentioned earlier, pH meters, titrators, particle counters. You've got what we call a long tail of PC-based instruments that probably don't even have APIs, but they produce files, sometimes proprietary files, sometimes standard CSV, ASCII, PDF files. Then you have enterprise systems like enterprise chromatography. As mentioned, I came from Waters, was there 36 years, was the first field person in 1986 when they started connecting HPLCs to computer systems. Now that those are enterprise applications, you have streaming data from bioreactors. And then you've got the typical list that I've heard many people talk about at this event, your MES, your LIMS, your ELN vendors, your analytics tools, data lakes, which are data aggregation tools, but could be an endpoint rather than a starting point for getting data moving. As I said, connecting two endpoints is not difficult. It's been done for probably 20, 25 years. You can connect a balance as a standalone instrument to a LIMS system. In and of itself, it's not a challenge. But as you connect more and more things together to start to produce a lab that's functioning, you end up with what we call a subway slide, where you have all of these point-to-point configurations that are brittle. Anytime there's a version change or an upgrade to an instrument, it needs to be re-engineered. Typically, it's hard-coded in how that workflow works. And there's not a lot of flexibility in moving data around. If you wanted that balance, let's say, to send its weight to the chromatography system for quantitative analysis, that would be a challenge in this environment. Typically, it would be directly connected, for example, to the LIMS system. So moving any piece of data in multiple directions concurrently has been a challenge. So the platform concepts that we've introduced are more about peer-to-peer networking. And in a peer-to-peer environment, each endpoint has equivalent functionality to every other endpoint, whether it's a simple device like a balance or a complicated PC-based instrument or LIMS or an MES. They both can send and receive data. And the magic of this is to do it in a manner that allows that data to be reshaped and restructured depending on how the endpoint it's connected to needs to consume it. So having the ability to connect these endpoints in a manner that allows ultimate flexibility of where the data needs to go, what shape it needs to be in, and when it needs to be there, creates an on-demand environment for data. And really, this is the principle about having data mobility and access to data instantaneously. So some of the pillars of this technology would be the ability to connect. And I've had several conversations today and last night around this, because I think there's a perception that it's historically rightly so, connecting endpoints can be a challenge. You've got, I heard black box devices, right, that may or may not have access through RS-232. You've got API-based instruments, such as chromatography data systems and the applications. The key, though, to data mobility is to get all the required endpoints, no matter what their capability is, connected. So it could be benchtop devices, standalone PCs, informatics systems, and the use of web services where and when applicable. You're never going to have a homogeneous way of integrating. And having worked for Waters for 36 years, I know the instrument vendor mentality. They always want to differentiate their technology by having proprietary binary formats. So in order to get data mobility, you need a strategy that's going to allow to connect that instrument no matter what its output format is, and have that ability to reshape it. So the assumption should be that you can connect whatever you need to connect to and not leave anything behind. Any application, any device should be connectable. Once it's connected, then it's around orchestrating and moving that data. That's really the magic of data mobility, is orchestration capability that drives the lab automation. It could be event-driven. There are notifications along the journey of the data. Business logic can be applied to transform the data, getting back to that example of connecting a simple balance. When a balance is connected, you may want to truncate, you may want to average weights, you may want to round up that data in flight. So when it gets to its endpoint, it's in the right format. But track that so that you have a DI-compliant audit trail of how that data was altered along its journey. And then, of course, there needs to be monitoring tools to ensure that everything is flowing correctly. And another attribute is not to introduce an application. The scientists are already burdened with a lot of applications. Think of a real integration platform as a digital plumbing strategy that once it's implemented, it fades into the woodwork. And your applications that you use routinely, your LIMS, your ELN, your QBD, all the app will drive the workflow. They'll initiate the workflow, or the instrument. And that mobility happens in the background. So I mentioned earlier, we weren't ready to do a specific case study. And what I can talk about publicly, because it's in the public domain, is the work we're doing at Pfizer. Pfizer in February of this year presented at the BioForum and introduced a concept many of you may not be aware of called digital decoupling. Digital decoupling is being born out of decades of big pharma and biotech building monolithic centralized LIMS systems. And out of necessity, as the LIMS vendors introduced new technology, like environmental monitoring or instrument integration or metrology data or analytics, They kept building these monolithic limb systems only to find they were now stymied and not agile to move quickly and shift. So this digital decoupling notion has been happening in the fintech space, the financial services space, for the last several years. But now it's starting to show up on the lab bench. And while digital transformation has happened in the back offices of many companies, the last 5 years I was at Waters, they went through this big digital transformation. We haven't seen digital transformation happen at the lab bench, really. It's been slow to happen at the lab bench. But this digital decoupling we're starting to see at more and more big pharma and biotechs, putting best-of-breed solutions in place to solve whatever the problem is. Could be environmental monitoring, instrument integration, LES, right, or ELN instead of having it built in, and then just expecting that they'll work together. And that's really what Pfizer's doing. Pfizer presented the notion of digital decoupling, as I said, at BioForum, and this article was published recently around their use of data mobility technology to connect about 7,000 instruments across their global manufacturing fleet. So next time I see you, I'll be sure to be wearing maybe a lime green suit to pick up the color of our logo, and maybe even talk in more detail about how Pfizer is using our technology.
Yeah, I'm here to talk about a little bit more about philosophy, but also to tell some stories about my experience working in the CMC arena and how I've come to believe that knowledge management is really the key to effective risk management. The key ideas I'm going to talk about is knowledge is what we need to have better organizations and better processes. And knowledge management is essential for that. And risk management is incomplete without effective knowledge management. There are three main dimensions of knowledge management that I'm going to talk about. This may not be a complete model, but this is the way that I view it. One is discovering new knowledge, and that's mostly the area that I've worked in, and is what for me is fun, is learning things and understanding systems and how things work. But then a real important component is once you've discovered it, you've got to share it. And then finally, you've got to make it available to a broader area. And altogether, those form a triad that is knowledge management. So the foundation of that of knowledge is theory, right? You got to know some basic science and engineering to be able to do CMC work. I mean, that's just a given. And, what's the knowledge is power, right? So knowing things helps you know how to know more things. And of course, prior experience too comes in a lot. So, if you're developing a new MAB process, having worked on one before really helps you. Or being able to learn from those who've done it in the past too, and not having to relearn things that they had learned. But in addition to knowing things, you also have to explore and ask questions. You have to have hypotheses, and you have to ask what if. One of the real key aspects that I always tried to push forward when we were developing process is expanding the boundaries of our knowledge. Not just focusing on what we think we know, but also understanding what we don't know. And finding that boundary and maybe pushing beyond that. That requires humility. It takes a lot to admit that you might not know something, especially if you're the one who's the person that's responsible for developing new process or understanding the science and being the expert. And I think one real key way to get past the pride and the fear of going beyond what we think we know is using structured approaches to gaining knowledge. I'm a big proponent of using experimental design. And I've had a tremendous success in my career using experimental design and working with others to uncover new knowledge. Other modeling approaches also are reasonable. Some, in silico modeling is really important too, as long as you have the good theory and good science to back it up. And then finally, we miss this a lot sometimes, is confirmation. Right? We do the study, we think we know the answer, but does it really repeat? there's a whole crisis in science right now called the confirmation crisis or the repeatability crisis, where people will do a study and they'll publish a study result and someone else will try to repeat the study and they can't confirm the result. And really that's just a statistical thing. It's what we call a type 1 error, right? There's going to be some things that look important that really aren't just because of variability. And so designing good studies and good approaches for generating knowledge and then confirming what we know is really important so that we can make that knowledge lasting and useful for a long time. So once we've got to where you've generated knowledge, it's really not useful unless you can share it with somebody else. I used to say a lot, if it isn't recorded, it didn't happen. And we're not just talking about compliance matters. We're talking about if you didn't write down what you learned, if you didn't share it in a way that other people can understand it and consume it, it really was not a worthwhile activity except just for you, maybe feeling good about yourself. I also feel like a real good key to doing this is to do it visually as much as possible. I think there were some other talks earlier about how do we have tables of data everywhere, but do we really have knowledge? Well, I'll show you an example of what I mean by that, but having a graph, putting it in a picture tells the story. And actually, ultimately, knowledge sharing in many ways is storytelling, right? It has an emotional aspect to it. You have to connect to the heart and the mind to transfer knowledge effectively. That means keeping the person who's going to consume the information in mind. And you may have to frame or write or present the knowledge in different ways to different types of people. Imagine how you were going to write a process description for somebody within your company that's going to have to do the tech transfer and stand this up at a new site versus how you might write it for a regulator who's going to have to review your process and approve it or not. You might write those differently because there are different audiences and there's different goals that you have for that knowledge transfer. And of course, standardizing as much as possible where it's appropriate is helpful. I've heard a lot of talk about that today, and I think that's really good. Mainly it's around calling the same thing the same thing. When I was doing some work at Merck, we worked on a big project, and a big component of that is we just standardized what we were going to call all the quality attributes. We're going to call them the same thing for every step, for every project. And what that enabled us to do was then to build standardized tools that people could use to do analysis, right? Because if the name was going to be the same every time, then we could know what we were expecting to get, and we could then write statistical analysis tools and data visualization tools and reporting tools that just worked. And it took all of that cognitive burden and hassle off of the scientists who were having to actually do the analysis work. Of course, templated approaches help too. Having data templates and how you want data stored and recorded is very helpful. And it makes me wonder, do we need more data standards in the CMC area? That's a real challenging aspect. The FDA tried several years ago to push a standard out for stability data. And it really didn't go anywhere. And they pulled that back because there were just too many challenges, too many questions, too much pushback from industry. But I think we need more data standards or data standard approaches in the CMC area because that enables tools for all of us to use. Companies like QBD Vision can build tools that everybody can use without a lot of extra customization when we do that. Here's an example about talking about putting it at a table. So this is data, right? This is an experimental design that was done to optimize a reaction. And the goal here is to maximize the reaction completion. There's all the data. It was a fairly extensive experimental design. There's the summary of the analysis. The factors that we looked at were the amount of catalyst, the temperature, and the concentration. And all of them are statistically significant. Woohoo, we got the answer. But how much knowledge do you have, right? Compared to this approach, how about putting your data into a graph? When I was working with a research group, when I was working at Elanco Animal Health, they'd just newly been acquired by the company, and they were very much in the let's have a table mode, and let's have tables, summary tables. And I said, well, give me that data and let's make a graph. And so I started this mantra, whenever you have data, you should make a graph. And the people got really tired of me saying that. Whenever you have data, you should make a graph. But then they started to say it too, which was really fun. Anyway, so this conveys more about what's going on. That graph that's on the right is a summary of the experiment in a picture. I don't really need to tell you much more about what I did in the experiment. It's all there. And then what's on the that's on the left. On the right is the prediction profiler. It's a tool we have in JMP, which is an interactive way to see when we fit a model to the data, what's the effect of the different factors on the reaction completion. And you can even couple that with a simulation. If you know the variability of the input parameters, you can see how would that transmit into the output of that model and get an estimate of risk. What's the chance we're actually going to be able to meet our goals or our specification? Okay. Finally, I'll talk about knowledge curation. I mean, we have lots of knowledge, but where is it? Right? And a lot of people have talked about that before, so I'm not going to dwell on this too much, but we need structure, we need data that's easy to find, easy to search, and easy to combine. And we need experts who know good and good tools to help us with this. So maybe we need knowledge librarians. We used to value that as a skill, the librarian. Do we still value that? Or do we all have to be librarians now? Maybe that's the answer too, I don't know. And we need good databases and search tools. There's a reason why Google is a verb, because it's pretty good at getting us the information we want when we want it without us having to do a lot of extra work. So risk is mostly driven by what we know and what we don't know. And the key to effective risk management is good knowledge management by developing good knowledge that we can apply to our processes so that we can have good control strategies. And without knowledge, we default to fear, we default to politics or pride and hubris, or we just default to let's just do it the same way, just like we've always done. And we lose opportunities for innovation. So if you don't know, discover new knowledge. If you know but others don't, share the knowledge. And if you want others to know what you know, now and in the future, curate that knowledge. I'll finish by telling some war stories. And I wanted to spend a lot of time on this because this is the emotional thing. I wanted to make a connection with you. And this were some significant events in my career. So talk about some of the good and the bad and the ugly. So this was one of the ugly situations I worked in. So I was a new process engineer, and I was working on a formulation problem in the process. Basically, the process was a liquid intermediate. It consisted of taking an incoming pure API, doing some additional purification on it, doing a solvent exchange from organic to aqueous after we did an extraction, adding a preservative, and then packaging it. Pretty simple, right? Not that complicated. But it had this really complicated intermediate step. So what we would do is we had these two in-process checks. When we were adding the preservative, we'd add 95% of the target amount that we thought we ought to add. And then we would sample and we would measure the content of the active and the amount of the preservative. And then we would say, okay, how much more preservative do we need to add? And we had a formula on the manufacturing instructions that the operator had to get a calculator out and do the math with. Okay. And then they'd add the additional amount of preservative. Then they would sample again and check that the amount of preservative was what we thought we added. And then we would go to packaging. Why did we do that? I mean, it's crazy. This actually added, for a process that took about 7 days, it added 2 days of cycle time, right? Because of just waiting for results to come back and doing the calculations and all this stuff. So why did we do it? Well, the reason we did that was really because we didn't have any knowledge. We didn't have knowledge curation. Actually, there was knowledge in the past, but no one knew why we did it anymore. I actually asked the question, why do we do this? And everyone kind of went, well, that's just the way we've always done it. Ended up doing some digging, like actually going and finding old laboratory notebooks from the archive and digging through it and realizing, oh, here's what happened. One time we had a problem, somebody accidentally added too much preservative and they over-formulated it, right? And you know if that happens, you can't add more active in, right? Because that's bad, that's not GMP. So we can't adjust by adding more active in, so we had to burn the batch. They had to destroy it. And somebody in management said, that is never going to happen again. You need to make sure that never, ever happens again. So they said, well, we'll just do it this way. We'll just be cautious. We'll add a little bit, and then we'll adjust. You know what that actually did, though? It actually made the process worse. You can actually show mathematically, when you're doing this formulation process, if the measurements you were making to check and adjust have variability, that variability transmits into your final product quality estimates, and twice. You pay the price twice. It actually doubles. Whatever your measurement variability is, you get twice as much in the output. Okay. So how do we fix it? Through gaining knowledge and using appropriate risk management. We actually gathered some data. We got that old historical data. We did some really statistical simulation modeling. We actually looked at the process. Over the time, we'd actually done a lot in the process to reduce many of the risks, almost all of the risk to misaddition of the preservative. It was much more precise and much more automated. So we said the risk is zero now that we're ever going to do this. So let's just get rid of these in-process tests. So we ended up just doing it where we said, just add the amount we think should be there, check it, and then go. Right? That's what we did. So we removed three of the 4 in-process tests we were doing. We removed a day and a half of cycle time. That increased the capacity of this process by about 30%. Just that. Okay. Oh, and that took about 6 months to justify and get it done, too. So it was a significant 6 months of my life. Here's another one. So now I'm in a different role. I'm the lone statistician in CMC development at Elanco Animal Health. And we have a new drug product we want to get registered, and we're writing a tech section, CMC tech section. It's for a tablet that we're making for dogs. And the data was just a mess. We had data, but we didn't have knowledge. We had data everywhere. It was not easy to get to. It was not easy to compile because things were called differently every time. You know, like dissolution was called disso in one experiment, and then it was called dissolution 45 because it was the dissolution taken at 45 minutes, and then it was called dissolution with a %. You know, just crazy stuff like that took so much time just to fiddle with to get the data ready. So the data was a mess. Actually, the data had errors. It was a lot of it was like unverified or hadn't been recorded properly or carefully. And the process knowledge associated with all that data was just not well reported or communicated. This was the same group that was not using, that was not using graphs a lot, so it was a lot of tables. So they actually weren't looking at the data to see if there was real issues. So we spent so much time just finding the data, documenting it, verifying it, finding errors, rechecking it, fixing the errors, writing, and that led to writing and rewriting the tech section because we're saying we're writing one portion, like when we're focusing on this process step. Where's the data? I'm not sure where's the data. Let's go find it. Okay, we write what we think it is. Oh, the data's wrong. We got to redo all of our graphs and our tables. So it made that process much, much longer, very frustrating. And this was the last thing we had to do before we were going to go to submission. Right? It was the last thing that we needed to do to get approval. Because in animal health, I heard someone say that CMC is not really a problem for the timeline. For human health, that's not the case for animal health products. The clinical trials in animal health are easy and they go fast. CMC is always on the critical path. So anyway, but we got that, we worked through it, and then we made some improvements. We said we don't ever want to go through that experience again. So we did some work on standardizing and making that better. Here's just some general case examples of what I call the DOE Resistance. So the one was a formulation selection issue. So, you know, the key to having a drug product is having a formulation, and that's usually the first thing you've got to decide. What's the formulation I'm going to use that's going to be stable and manufacturable? And this scientist was responsible for defining the formulation, and she had done many, many studies and hadn't really come up with a formulation that anybody thought would work. So someone said, you need to go talk to Sam, have him take a look, help him, let him look at your data, see if you can figure some things out. And so she brought me your data, we looked at it, we brainstormed, we made graphs, And there just wasn't a lot of information in her studies because she wasn't really exploring a lot of options. She was doing this one factor at a time and just kind of nudging one way or another, not using an experimental design approach. And I proposed to her, I said, well, we've got a lot of general information. I think we could probably design a good experiment. Why don't we do an experimental design? It might take, I was saying like 20 to 30 different formulations, new formulations. We'll do an accelerated stability study, and we'll probably get a good idea what the answer is after that. She said, I'll think about it. I don't think I need DOE. I think I can figure it out on my own. So a year later, mind you, she comes back to me and says, my boss says you need to help me because I still don't have an answer. And so when she came to me the first time, she had done about 50 formulations in 3 different accelerated stability studies. Guess how many more formulations she had done in the interim between the first time we talked and then when she came back to me again? About 80. 80 more formulations. Remember, I told her we could probably do 20 or 30. And I told her, I said, if you'd have told me a year ago we could do 80 formulation experiments, 80 different formulations, we would have knocked this out of the park. I mean, we would have the answer now, and we would have had it earlier. So anyway, you probably can figure it out yourself, but you don't have time. And that's the moral of the story. You got to get the answer, and using experimental design is a good way to do that effectively and efficiently. The other one is, this is another one where it was, we don't have time to do DOE. We were characterizing a tableting process, and it was a little tricky. It was going to be hard to meet all of our critical quality attributes, that targets and ranges. It was going to be hard to control. The formulation maybe wasn't quite the best, so it was very sensitive to compression and how much you compressed it, and that would radically change the dissolution of the tablet, and that was the key issue, was targeting the right dissolution profile. And we had done about 4 full-scale production runs, and it turns out lots of times with drug tableting, full scale is where you really got to do your experiments, because that is the system that models the process the best. The small scale runs don't always model the full scale system well. And so someone said, and this again, we're on the critical path, we got to get this solved. And I said, well, we think we know what it is. It's these 3 factors that might impact the tablet compression, or the dissolution and tablet compression. And we could do a set of experiments. It might require us about 8 different batches. And everyone's eyes got really big. 8 full-scale batches? Well, yeah, well, that's what it's going to take to get the answer based on what we know. At least that's what I thought. And everyone said, we don't have time to do that. A year later, guess how many more batches we had done and still didn't have the answer? 12. 12 batches. And I remember, and we did muddle about, and by the end of that, we kind of had close to an answer. So it worked, but it was not a great process. And that process was going to be hard to run and control. I told that story to our vice president about 6 months after all that, and his eyes got real big. And then he was like, everyone, you need to use a DOE. You got to use a DOE. You got to use a DOE. And so that changed a lot of things. You don't have time to not do a DOE in many cases. And here's a good example. I know I'm running over. I'll finish up. So this was just a new synthetic route for an API. It was late-stage development, but someone said, I've got a new way to make the API. It's going to save us a lot of money. It's going to shorten the cycle time. We don't have to build new equipment. We don't have to buy new stainless steel tanks. It's going to work. So the development team says, okay, let's go for it. And we had to develop the control strategy of this really quick. And the whole team bought into using experimental design. And that whole process worked wonderfully. Because we generated knowledge effectively and efficiently, we could predict what would happen. As a matter of fact, when we went into validation, we predicted almost perfectly what the outcomes of that validation run would be, because we could predict the reaction kinetics, we could predict what would happen during the crystallization, we could predict what would happen during drying, we could predict what would happen during milling, we could predict what that final product quality was going to be. And it worked great. It really led to a quick process development, and in that process later on, which by the way made billions of dollars for the company. It was a big product. And delaying that would have meant real dollars. You know, if you sell $1 billion a year and you delay a month, you can do the math on that, right? It's over $100 million. So that was a real good success story, one of the key highlights of my career. All right, so wrapping up, you can't manage risk without knowledge. You can't truly have knowledge unless it's discovered, shared, and curated. And the key to success in the CMC technical arena is effective risk management, which is enabled by effective knowledge management. Okay? Right. And there's my contact information. Happy to talk to you about what we have to offer. For JUMP, I focus on the needs of folks in the life sciences arena, and we're really kind of on the knowledge discovery, knowledge generation area of the spectrum of what I was talking about. But yeah, I'd be happy to get connected with you. Thanks.